Hello, I'm Gunther, and I'll be presenting on Gromov-Wasserstein for protein structural alignment, joint work with Patrick Nicodemus and Pablo Camara. Protein function is largely determined by its three-dimensional structure. For example, here we have a protein bound to a ligand at an active site. This is generally more important than its sequence and better conserved evolutionarily. There's now extensive data on protein tertiary structure, with hundreds of thousands of proteins determined experimentally and hundreds of millions predicted. From this, methods for quantitatively comparing proteins are key to forming new insights from this data. We start by defining the Gromov-Wasserstein, or GW metric, which measures morphological similarity between two objects using a weighted coupling, denoted T, between points in one object and points in the other, which minimizes total distortion. Here, for example, we have a coupling between two human figures. It connects body parts in one to the same body part in the other, thus minimizing the distortion. For two proteins, the distortion between pairs of alpha carbons in one and pairs of alpha carbons in the other is defined as the difference of intramolecular distances. So we see here xi and xj, two alpha carbons in one, we look at the distance between them, and similarly the distance between yk and yl in the other, then the distortion is the difference of the distances. Gromov-Wasserstein then measures the total distortion, so the sum of the distortion of all possible pairs times their weighting, and then we look at the minimal one over all couplings. In our work, we developed variants to the basic Gromov-Wasserstein that can account for isoelectric points or other biochemical data. We can have it give greater weight to local distortion over global dissimilarity, and it can incorporate sequence alignment. In our work, we studied riboviral RNA-dependent RNA polymerases, RDRPs. Riboviruses are a realm of viruses, including, for instance, COVID, which include many human pathogens. RDRPs are key enzymes in the replication machinery. In this picture, we have three examples of RDRPs, and we see at their active site there are three highly conserved motifs labeled A, B, and C. We know that they can appear in different sequential orders, both ABC and CAB. To benchmark the performance of GW, we ran it on a dataset of 4,615 computationally determined RDRP structures and 512 non-viral, though structurally homologous decoy proteins, and compared it to the leading methods of TM-Align, Fold-Seq, and DALI. We note that GW is faster than all of the methods except Fold-Seq. In our benchmarking, we saw that GW did a good job of clustering the proteins based on either their phyla of origin for the RDRPs and distinguishing the decoys from the RDRPs. To quantify this, we made a 3 nearest neighbor classifier trained on the GW distances. For each phylum, we used 10-fold cross-validation, where each iteration we used a tenth of the phylum and a tenth of the decoys for testing, then the proteins from the other phyla and the remaining decoys for training. The goal of this was to simulate a real-world scenario where the proteins you're interested in are not closely related to proteins you already know about. For each phylum, we then took the Matthews correlation coefficient, MCC, of each of the 10 tests, then averaged them. We see here that overall GW and the other methods have rather similar accuracy on average, but the key takeaway here is that GW is much more consistent across the different phyla. The next task was to try to determine the active sites of the proteins based on stress. So for this, we start by defining the stress of residue based on its contribution to the total GW distance. So sort of how much distortion comes from a particular residue for a given pairing. For 97 RDRPs, we computed all pairwise GW distances and the stress at each residue for each pairing. This gives us a way to identify the structurally conserved regions as those with the least stress. For instance, here we have an example of RDRP, where in the center, near the active sites, which are highly conserved, we have much less stress, indicated by the bright yellow, whereas further away from the active site, it's much less conserved, so we have much higher stress. We can use the lower stress regions to predict the locations of the active site, in particular their A, B, and C motifs. For a given protein, we can vary the stress cutoff levels to generate a precision recall curve, and we did this for all 97 proteins. We then compared GWPro to other alignment methods and found that it outperformed them. As proof of concept, we used GWPro to identify circular permutations in the RDRP dataset. For instance, here we have two RDRPs which are highly structurally homologous, whereas there's much less sequence homology. In the first one, the C motif comes before the A and B, whereas in the second one, the A and B come first. For two proteins, we can generate a coupling between them, then use that to calculate the probability that the order of two amino acids in one is switched when aligned to the other. We can use this to infer the location of circular permutations between the two proteins. Most of this plot is very light, indicating that the order of almost all amino acids is preserved, with the exception of the dark rectangle in the middle, indicating that the C motif switches places with the A through B region when you go from one protein to the other. Thank you. We'd also like to thank Drs. Robert Edgar and Artem Babayan, and support from the NIH.